So, welcome everyone, and thanks for coming to our intertidal monitoring refresher training today. I'm Rondi, the Aquatic Reserves Monitoring and Stewardship Coordinator for Resources, and I'll be leading surveys at Fidalgo Bay and Cherry Point Aquatic Reserves. So this is just a quick slide with some reminders for Zoom controls.
Barbecue chicken, very good.
And plans for today. So as Eleanor mentioned, we'll be recording the training today. So if you don't want to be recorded, keep camera and microphone off. And if you don't want to be, and then pose questions in the chat. Another reminder is to please remain muted unless you're called on or in the breakout rooms at the end. And then if you have a question, you can put it in the chat or use the raise hand feature that you can access down at the bottom through the reactions icon. So this is today's agenda. We're part of the Welcome and Introduction Now by myself and Eric Blakey from DNR. Then we'll transition to going over species swaths with Betsy Carlson from Fort Townsend Marine Science Center. Eleanor, also from Resources, will cover COVID and intertidal monitoring protocol. Erica will cover beach elevation profiles and substrate. Eleanor and I will cover quadrats, and then we'll have time for Q&A and as well as breakout rooms by survey area. So we'll break it up into like Cherry Point and Fidalgo Bay, as well as Port Townsend area. So again, thanks for coming to the training today. We'll have folks from Port Townsend Marine Science Center, the Department of Natural Resources Aquatic Reserves Program, and Resources walking you through how to conduct surveys in 3 aquatic reserve areas. Also special thanks to our volunteers on the citizen stewardship committees who helped make this work happen, particularly for Fidalgo Bay, Cherry Point, and Protection Island. So what is community science? It is, these surveys are part of community science, and it's a vital part of our work as it lets us cover more ground and fill data gaps better than if we relied on only staff time. It's also a great way to raise awareness for environmental issues and get people involved in the scientific process. We have protocols in place to ensure the quality of our data, including regular quality control checks by staff and lead naturalists, both on the beach as well as for data entry. It's also just a really great way to learn. Our staff and lead naturalists are available to help with questions on the beach, including species ID. I've heard from folks that it's really fun learning about the scientific process and learning, just about all the cool beach critters out there. And so we conduct intertidal monitoring specifically in order to get a clear understanding of the flora and fauna we can expect to see in a given area on the beach. And though through this, we maintain a long-term baseline inventory that we can refer against climate change impacts, track invasive species introduction, track infections such as sea star wasting, and assess restoration success. There's so many different reasons to monitor, particularly within aquatic reserves. And here are some examples of why we monitor. For example, we can monitor for the spread of invasive European green crab. These are highly invasive crabs that have been found in areas near several of the aquatic reserves we monitor, and they have the potential to cause widespread destruction in several critical habitats such as eelgrass beds. So very important to track. We could also assess environmental and biotic conditions after an oil spill, for example. Several of our sites are near oil refineries or in areas that see a lot of vessel traffic, and a spill could have pretty serious impacts on our intertidal areas. And so we monitor several sites each year. Some are monitored for enhancement or restoration, as well as some are just long-term monitoring sites in the aquatic reserves. Typically, we monitor about 2 sites per aquatic reserve, sometimes more depending on the year. And so I've been talking a lot about aquatic reserves, but I haven't really explained them until now, so many of our sites are in aquatic reserves, which are protected areas managed by the Department of Natural Resources. particularly the Aquatic Reserves program. These are areas that were established to protect and restore native habitats and natural processes that are important for a healthy Salish Sea. And with the help of groups such as citizen stewardship committees, the reserve network promotes stewardship and understanding of aquatic habitats and species. And one thing that you can actually do, I mean, beyond participating in our surveys, one thing that you can do to support the reserves is to fill out this pledge in the chat. I'll be putting it in. Oh, Eleanor beat me to it. So this pledge basically helps us meet grant requirements that ensure there is an AmeriCorps filling my current role in helping make surveys run smoothly. It's part of our way to communicate to AmeriCorps to say, hey, we're actually meeting our requirements and getting people engaged. So if you could take this pledge after the training, that would be really awesome. And I thank you ahead of time. All right. So Cherry Point is one of the reserves that we monitor for. It is the northernmost aquatic reserve, and it's located in Whatcom County. This year we'll be surveying 2 sites, Point Whitehorn and Intalco. Just a heads up that our exact schedule is still being worked out, as some sites may change, but this is overall our plan for the season. And we'll be sending out the formal schedule later as well. Another aquatic reserve that we monitor in is Fidalgo Bay. And this is located in Skagit County near the city of Anacortes. And this year we'll be surveying 3 sites, otter, fur, and trestle. For fur, we'll also have a group of students from the Anacortes High School helping for part of the day, which is really fun and exciting. And so now we'll transition to Betsy Carlson from Port Townsend Marine Science Center. And Betsy,
hi, everybody.
if you want, feel free to tell me when to move to the next slide.
Okay, thank you. Thanks, Randy. I'm so glad to see so many familiar faces and new names on this. Our sites that we work with in the Port Townsend Marine Science Center and the Aquatic Reserve, Protection Island Aquatic Reserve, is at Cape George site and Kinsey Beach, which is actually in Fort Worden State Park. So we'll be working there this summer in June. Next. So I know a lot of you are involved with this kind of work because you are caring about it and you want to learn more. We just want to remind everybody of some things to keep in mind when you're out at the tide pools or out on the beach. Just keep an eye on watching where you put your foot. The rocks can be slippery, with any kind of marine algae on there, but also that's where the things we're out there to look for and to count and keep an eye on, that's where they live. So keep your eyes open where you're walking, and keep your eyes and ears open for what's happening in the water nearby. Listening to the waves and listening to the shoreline, it can kind of sometimes can surprise you and can also make it a little slipperier if the water washes in where you're walking. So just be careful for that. And get yourself into a Zen mindset when you're out in the beach. I think that's a good way to think about it, because we're looking carefully for teeny tiny little things, and sometimes they hide pretty well, and sometimes they're hard to figure out. And if you're just patient as we look, you'll see so much more, and you could learn a whole lot more too. So be patient when we're there. And really important is, if you're picking up anything, just to put it back exactly the way you found it, and very carefully, because that's somebody's home and you want to protect their home for the rest of, as long as we can. Take pictures, leave only footprints. That's just a good way to function in the world, in my humble opinion. And if you're going to touch anything, touch it gently and just briefly. Good idea to wet your fingers before you touch anything, 1 or 2 fingers and just let the critters have their space. But sometimes it is really fascinating to figure out what's there. And we're there to conserve and inspire, and you'll have the opportunity, perhaps have the opportunity to even talk more about what you're doing with other people. So anybody visiting on the beach, like, this is a pretty cool project, and you get to share why we're there. Right. Next. So to remind those of you who have done this before, and to inform those new folks, if there are any, there are many different parts to the intertidal monitoring project. These are done once a year, usually at a good low tide, and it involves several different aspects that we are measuring and counting and looking for while we're there. There's a species swap, the beach elevation profiles, and the quadrats, and I'll get to more of those in a minute. And then cores, only Fidalgo Bay site does the cores, and that's to get down and look for benthic organisms within below the surface. So that's all. And okay, so I'm gonna talk to you now about the different parts. When you head out to the site, it may or may not be laid out. So that blue dot is the starting point, and we all have preset starting points at the same place we try to get back to year after year, and everything else goes from there. So if you're wandering out there and you know see where the starting point is, it's a good idea to just plop your backpacks and your other stuff distance away from that, so we're not trampling the area that we're trying to measure and look for. And this is a general idea of the whole layout, and I'm going to talk through the different elements of it. So let's go to the next one. Our profile line is the center line. It goes from the shore, the starting point, down to the water. And along this line, we use this to get a sense of the structure and the slope and sediment aspects of the beach. And this line is right in the middle. And if you know approximately where that is, you have 33 feet on one side of that line and 33 feet on the other. So it's a whole area that we're looking at is 66 feet wide. And if you think about how tall 33 feet is, that's as tall as a telephone pole. So when you're walking up and down, you'd want to be outside of the whole area as much as possible. So just keep that in mind. It's a good distance from the center line. Okay. And the other, so that purple line is profile. These are the transect lines. The transect lines run more or less parallel to the water or to the shore. And it's from these lines that we measure the quadrats, or we lay down the quadrats. And there are 3 different transect lines. And then we put 4 quadrats on each of those lines. It's good for us if you're walking in and out of these sites to walk on the land side of that transect line, because then you won't affect you won't step on what we're trying to measure in the quadrat area. And yeah, we'll get to the quadrats next. Okay, these lovely green squares, which are actually white plastic things with grid lines in them, and you're going to hear much more about that in a moment. There are 4 quadrats that are laid along the transect lines. The upper set is +1 tide, which is 1 foot above the 0 tide, and then there's a set of -1 quadrats that we also do. The quadrats, it's a good idea to try and get to the ones that are closest to the water first. So if the tide starts coming back in while we're doing it, there's more time to get to the other 2 layers. So start with the ones down by the water. And please walk either outside of the whole area or on the water or land side of the transect lines. Okay, and let's talk about swaths. So not only do we get a subsampling with the quadrats of sort of abundance, we also want to know what is there, what different organisms actually exist in this 66-foot wide by however long your sampling area is. And each of the swaths are measured by or visited by a naturalist, the lead naturalist, and the scribe. And they're looking for whatever living organisms are found in there above a certain size. And so that whole swath gets done bit by bit. But if you're doing a quadrat and you see something really fascinating, it's always good to let a lead naturalist know, because you might see it and it might hide when they're coming through. If you're interested in being a scribe for the lead naturalist, it's a pretty busy job, but it's also a great way to learn about species identification. And I believe that's all I got to say. So I'm passing it on to Eleanor.
Great. Thanks so much, Betsy. And it's also great to see some familiar names in the participant box and some new ones. So welcome to those who are new and welcome to those who are returning. My name is Eleanor Hines. I'm with Resources. And so I'll be helping Rondi out with the Fidalgo Bay and Cherry Point surveys. And so we wanted to just real quickly touch on COVID and Intertidal for 2022. As more folks have become vaccinated and restrictions have lifted for COVID, we're also similarly lifting or relaxing a lot of our restrictions. However, next slide, Rondi, please. However, we will still be asking that you keep your masks handy, and as possible, physical distancing is something that we should still try to do our best at, just to keep everyone safe and healthy. We don't require at this point in time folks wear their masks, but we do ask that you have them on hand, because it's easier to just be in the habit of bringing it with you everywhere than because we don't know what's going to come in the future and things keep changing. So if you have any questions, we do encourage you to ask at any time. You can ask myself, Rondi, Erica, or Betsy. If you're unsure, we do still ask that in addition to keeping your mask handy, that you do stay home if you're not feeling well, or if you've been around somebody who has tested positive for COVID. Vaccinations and boosters are great, and anyone is welcome to wear a mask at any time. So if you, for any reason, feel better about wearing a mask, that's great. We definitely welcome people wearing masks, even if we don't require them. And again, if people have any questions, please ask at any time. And next slide, please. So to wrap this section up, I'm just going to highlight some of the general safety and expectations, because COVID isn't the only one out there. So just a reminder to please dress for the weather. And it's oftentimes a good idea to bring layers, sunscreen, hat, rain gear. Sometimes it looks beautiful and sunny when you set out for the day, and then all of a sudden a dark cloud rolls in, or vice versa. And sometimes also on the beach, there can be a breeze. And so while it might seem a little bit warmer at your house, it might be a little bit cooler on the beach. So just something to be aware of, and layers are usually a good idea. Do bring food and water, whatever seems like will help you get through the day. However, some of the sites do have limited or no access to bathrooms, so please plan ahead. Wear proper footwear and watch your step on the beach. Some of these beaches can be quite tricky to navigate. It's not the same as walking, down the sidewalk. There's a lot more obstacles there where it might be slippery, or there might be boulders and cobble to navigate across. Gloves and or knee pads are something that folks are definitely welcome to bring. We do have knee pads that we will provide. take your time and have fun. There's no reason to rush through this. And having fun is, I think, a really important part of community science. So if you're not having fun, take a moment, take a deep breath, take in the scenery, and remind yourself of where you are and what you're doing. And it's all about having fun while we're out there collecting data. And a quick reminder, I think Betsy did a great job of highlighting this, but to try not to walk through the survey area so that we don't trample that whole area before we get a chance to survey it. This especially makes a difference when we're at muddier sites. It becomes a lot more evident when you put down your quadrat and you see boot prints right through the middle of your quadrat. And I think that's what I have. And I think Erica is up next on the beach elevation profiles. Erica, are you ready?
I'm ready. Can you hear me?
Yes, I can hear you.
Okay, let's work on the slides next.
All
Can you
right.
see my screen?
It's coming. Yes, it looks great.
Awesome. That never happens. Okay, so I am here. My name is Erica. I'm here to talk a little bit about why beach profiles are important and to review the survey methods. So this is a part of the protocols that provides really valuable information about a monitoring site by measuring two really important and often overlooked factors if you're just walking along the beach, and that is the beach profile or the beach slope, and then also the type of sediment that exists on that slope. The beach slope, or how steep that beach is, tells us a little bit about what sediments are likely to be present there. And wave action on steeper slopes tends to wash more sediments away and leave us with kind of some coarser sediments. And sediment size is really important because it essentially decides what's going to grow on a beach, or what type of organisms are actually going to live and use that beach. So if you have sandier sediments or muddier sediments, you'll tend to have grasses like sea grasses that grow in the area. And if you have rockier beaches like this beach you see here in this slide, you'll see that it's kind of covered with algae and kelp. So, these are two of the most common types of vegetation that you'll see on beaches, and they bring different animals to the beach to use those resources. Another thing that sediment really makes a difference for is how organisms directly, or animals, how animals directly use the beach. So there are, for example, there's a really important fish called the surf smelt that lay their eggs on beaches, but they are really picky and they really like gravel beaches or sand beaches. And so kind of noting what's there as far as sediment or substrate gives us an idea of how those beaches might be used. So that's why it's important. Now let's kind of go into a quick review of how to get this information recorded. So the way that this starts, it starts along the profile line that Betsy just outlined. You start at the starting point, at the 0 on the measuring tape at the top of the beach. And one person holds a pole and another person holds the second monitoring pole and heads about 10 feet down the profile line. And the very first thing that both of those monitors are going to do is to make sure that their pole is absolutely vertical, which they're going to be able to do because they each have a level attached to their fancy measuring pole. This is a little bit tricky. And so if in this photo, you see only 2 folks that are on each pole, but if you have more people available to help, I find that it's often really nice to have 2 people on each pole, one person to level the pole and make sure that it's vertical, and then another person to take the measurements. And if you have a lot of folks to spare, it's almost nice to have 5 people on this part of the monitoring, because you can have that 5th person record all of the data as they go. So once these poles are level, the high-tech system comes into play. And this first person who's at the 0 point and who's looking out toward the water looks through a hole that's drilled in the in the tall pole, and they're checking out their partner's finger, and their partner is standing on their pole, making sure that it's level, and they're moving their finger up and down on this pole. And that first person is going to tell that person to stop moving their finger as soon as their finger is in line with the horizon, which is where the sky meets the water. And what you can't see on this slide right here is that these poles all have measurements marked on there, which we will see in this diagram, which kind of can be overwhelming. So in the future, because this is recorded, you can just look back and use this as a brief review to read through, but don't worry about reading it right now. All I want you to focus on is the bottom of the screen. This eyeball represents the first person standing at the 0 point, looking through that hole in their pole. And they're looking at this point in their partner's stick, where the horizon overlaps with the stick, and they're taking their measurement off of these marks. And so what I want you to do right now is to do a quick practice. This thick mark right here on the pole marks 0, which means that if you see that mark overlapping with the horizon line, you are absolutely level with your partner, and no positive or negative changes in elevation have happened along the beach. But just take a second and note that if each of these marks is 1/10 of a foot, think about what you would write down on your data sheet as far as the number of change. And then maybe write that down in the chat. I am just realizing I don't think I can see. So maybe, Rondi, if there are some numbers in there, you can read them out.
Not seeing any coming in yet.
Okay.
Oh, someone put in -4.
Close.
4/10.
Yes,
4.
there we go. Combine those two together and we get -4/10. And this is, I think, the trickiest part of this type of monitoring, is that all of these numbers above the 0 represent negative numbers, and all of the numbers below are positive numbers. And so if you think about why that happens, if the second person is walking downhill and the first person is looking at the mark above the thick 0 point, it just shows that that person is lower than you. And so it takes a little bit of calibration before you get started, but once you're used to it, it makes a little bit of sense. So yes, -0.4 feet. Okay. The other thing we want to make sure that you're doing as you go along is capturing significant changes and obstacles. And normally when you're moving along, you're doing 10 feet chunks. You walk 10 feet, you take a measurement. You walk another 10 feet, you take another measurement. But if you run into something weird, like a log or a boulder or a sinkhole, I don't think you'll run into a sinkhole, but for some reason, this is the photo we have here in our slides. What we want you to do is to stop and take a measurement right before that obstacle, right on top of that obstacle, and then right after it, so that we can kind of capture that in the data set. And what happens with that data when we look at it later then is we can see that something was there. So you'll notice when we look and we graph the data that you all gather, this beach was nice and gently sloped, and there was a huge log at right about 50 feet down on the beach. And the way that we know there was a huge log is somebody noted it on the data sheet. And so this is what a data sheet looks like for the beach profile. And over here on the right, you can see that they wrote exactly where they saw the log. And I think Jason made a fancy animation. So this is just kind of a zoomed in vision of what the data sheet looks like. And as you go down the line, each row is one data point. So if you stop at the 10-foot line. Remember the first photo that I showed you with one gentleman standing at the 0, one woman standing at the 10-foot. You're marking down all of the information about that 10 feet that you're on right there. So you've gone 10 feet from the 0. This is the cumulative distance that you've gone. So you can see as each new measurement that you take, you add about 10 feet, unless you come to a log or something else that you need to measure. These 2 columns here are where you're going to add the elevation change, whether it be positive or negative, and then the number. And then this last chunk is really just presence and absence. Do you see certain types of substrate where you are? And it's just a check if you do see it and a blank if you don't. One thing to kind of make that a little bit more explicit. So this is your profile line. You're starting here and you're walking toward the water. And if this is you and your buddy on your profile poles, where you're looking for that substrate is the entire area between the 2 of you, and then over to the edge of the swath on each side of the profile line. So this entire area is where you're looking for substrate and noting if it's present or not. And then the last thing I want to talk about are the substrate types. Most substrate we mark just by size. Shell debris is a little different, and shell debris is pretty easy. And then it's just, if you see a broken shell, that's shell debris. And the rest are mostly by size. This, I think, is the hardest. The clay, silt, sand decision is tricky, and it's done a little differently depending on which aquatic reserve you're doing this for. And so what I recommend you do before you get started is to just check in with whoever is leading that site that day, and they'll let you know what to do about the clay, silt, sand conundrum. They're tricky in that they're similar sizes, kind of like a grain of sugar up to maybe like a grain of rice. But the clay and silt are fine particles that cling together and don't really allow water to percolate through, whereas sand is really coarse and it does allow water to leach through. So it's kind of a texture thing. And then this is easy. Gravel, the size of a pea to the size of a chicken egg. Cobble, the size of a chicken egg to the size of a basketball. And the last substrate type is a boulder. So that's anything larger than a basketball. And that's really it. One thing that's really fun, I think, because I've done these profiles a bunch, and one thing I find really interesting is that as you start higher up on the beach, you're going to probably see a lot more different size variety in the sediments that you're noting. And as you walk toward the water, you'll see that there's a lot less variety, just because there's water that covers that area more often, and the waves kind of do a really good job of sorting through that sediment. So just kind of something fun to watch out for as you're walking along. And that's all I have. I don't know how to stop sharing. You have to stare at boulders.
Go up to the top of the screen where you're sharing from. And there should be kind of alongside the green sharing, red stop sharing.
Aha.
There we go.
Got it. Thanks, Randy.
No problem. I'm going to start our quadrat section now. All right, how is that looking for everyone? Great. Awesome. Okay, so yeah, I'm going to start covering quadrats. And so some of this is just a refresher from things that Betsy or Erica brought up earlier, but this is a layout of where the quadrats sit in our survey site. We'll have 4 quadrats along each of the tide lines that we have set out, each of the transects. And as was noted, also it can help to do the quadrats closest to the water first to make sure we get them done before the tide comes back in, which has been an issue we've run into in the past. And so we also make sure that the quadrats are placed with the upper left corner on the selected point facing the backshore or bluff. So the left-hand corner of the quadrat is on the water side, matching the point that we're supposed to match it to. And so what data do we cover with quadrats? So we file it into kind of two main groups, being percent cover and individual counts. Percent cover assesses organisms that are hard or impossible to count individually. For example, things like barnacles, algae, mussels, and other things. And then individual counts are good for things that we see lower in lower densities, such as burrowing anemones, crabs, and fish. And also to note, individual counts are for animals that are greater than 3 millimeters in size, which that's pretty tiny, so you can capture a lot of different things. And so this is what the front page of one of our data forms looks like. At the top, we'll have information about the survey, followed by a section for QC checks. And so this is a way that we ensure quality and consistency in the data by having staff and lead naturalists do one last check before you move on to the next step. So for each check, a photo is taken and a signature recorded. We have sections on the data sheet for before and after ulva removal, as you can see kind of lower down when you get into the percent cover, as well as for each photo and QC check. And so ulva is a type of green algae that can, at some of our sites, completely coat the beach. And that kind of, like, if we didn't assess before and after, we would miss a lot of critters that, you know, are under or within the ulva. So we do a before and after. This way we get an idea for how much ulva there is while still being able to record the other critters that might otherwise be hidden. And so when you move to the after ulva portion, remember that the before counts transfer over. This is a little confusing at times, but for example, if you saw 2 crabs before you removed the ulva, so maybe they were on top of it, and then when you removed the ulva, you found 4 more crabs, you would have a total of 6 for the after removal section. And there's also a section to record percent of substrate present. And one other note is that Port Townsend Marine Science Center has even another section for coming back eelgrass. And so kind of unlike some of our other sites at Fidalgo Bay or Cherry Point, they have, you know, pretty substantial eelgrass meadows to my understanding. And so unlike the ulva, we do not remove the eelgrass. It's very important habitat. So what you do is you kind of comb it back gently just to look at what's underneath or maybe clinging to the eelgrass. Again, do not remove it. It's very important. I guess, Betsy, since you're on, is there anything that you'd like to add?
Oh, you did great. Just move it out and don't pull it out.
Awesome.
Yeah.
All right, and so this is what the back side of our data forms look like, and this is showing the individual counts. And so again, all individuals larger than 3 millimeters are recorded. And one additional thing that you might see on the back would be a section for core samples where we do, for each quadrat, we'll take a core sample with big PVC tube and kind of look for things that are hidden in the sediment. And we would only be doing that at Fidalgo Bay where the substrate is really soft. And so now we're going to talk a little bit more about identifying all the fun stuff. And one thing that's really important to remember is that, you know, go at your own pace, take your time, ask for help when needed. A lot of this quadrat work relies on species ID, which can be really tricky sometimes. Things don't always look the same in the field as they do in your ID guides. And so if you're ever, in doubt, feel free to ask a lead naturalist or staff member to help. And it's also important to go slow because we want quality data, which means, being thorough and like really looking at everything. And yeah, so when in doubt, ask questions and take your time. And so I'll start getting into percent cover now. So percent cover estimates, it's important to not overthink it. This is something where, you can spend quite a bit of time going over like, oh, is it exactly like 20% or is it 22%? There's some strategies that you can use to kind of make the process go a little smoother. But yeah, don't overthink it, use the tools available. And it also helps to do this with a partner. To where you can kind of each do your own count and then compare what you each got. And some of the tools provided would be, we have these little 1% squares called cheese squares, where it can provide a really nice reference for when you're just starting out. And then also really important, make sure that at the end, all of your sums add up to only 100%. We don't want 120% of anything. That's not what we're trying to capture. So yes, don't overthink it, use the tools available and make sure things add up to only 100% max. And so here's an example of why we do before and after ulva. The image in the middle is an example of what ulva would look like. And on the left is a photo from one of our quadrats in the past where the ulva almost completely covered the entire quadrat and you couldn't really see much underneath. And then on the right is an image of where the ulva was removed. In that case, there wasn't really a lot to see under the ulva, but sometimes there'll be just a huge amount of critters or huge number of barnacles, just a lot to count underneath the ulva. So it can be pretty important to do both. And again, at Port Townsend Marine Science Center, they have eelgrass. Some of our other sites might not be quite as abundant with this, but it's also important to know that there's two species that you could expect to see. Zostera marina is the native species pictured on the left, where you get to see the rhizome or the root structure that's typically underneath the sand. And then on the right is Zostera japonica, which is a non-native species. You can tell them apart by how the native species has wider, thicker blades and a nice rhizome structure under the sand. And then the japonica, the non-native, has thinner blades with less of an underground root structure. And it's maybe a little easier to tell in person versus in the photos, but if you have questions, feel free to ask a lead naturalist or staff person. And so even though ulva will probably be the main green algae species you encounter, there's a huge variety of others to look out for in your percent cover counts. ID sheets and naturalists are a really great way to help identify these, especially because seaweed color can't necessarily be the most reliable tell. They can be really differentiated by shape, texture, color, and where in the intertidal you find different types of algae. So this is just a good selection of a couple that you can expect to see, including ulva. And here's a few examples of brown seaweeds. So they tend to be fairly easy to identify as being brown. Typically they come in sort of a brown, olive color, or kind of a just darker tones. And so there'll be kelps, rockweed, or sargassum typically. One thing to remember about rockweed is that it can sometimes be a really nice vibrant green color, but it is still in fact a brown algae. And the rockweed is the middle photo. And once again, color is not always the best indicator, as some red algae can actually appear greenish, but for the most part, they'll have a strong red or blackish color to them. And this can even be tricky for some of our naturalists with the red seaweeds. So just to remember, if you're ever having a question or like confused or unsure, reach out to staff, refer to your ID sheets, and reach out to lead naturalists. And so getting into some of the critters, barnacles are one of the most abundant percent cover species we see. And it's really important to pay attention to whether or not the barnacles you're looking at are alive or dead. And this might actually be pretty important since we'll be evaluating post heat wave from last year. But we do not count dead barnacles. And you can tell if they're dead, for example, by whether or not they're empty in the middle. So in this photo, for example, you can see ones that have nothing in them, they're empty and they kind of look dark in the center. So yeah, we don't record dead barnacles, but the live ones are part of our percent cover. And again, sometimes it can be really hard to tell in the field, so feel free to call someone over if you have questions. And mussels are another percent cover species. They're fairly variable in color. They can look blue, brown, or black, and you'll often see them nestled around or in barnacle patches. We don't see them very often at most of our sites, but definitely good to keep an eye out for them. Next up, I'll be talking about aggregating anemones. They're really fun. And we count them as percent cover because they can occur very prolifically in certain areas to where it's very difficult to count each one. They come in like huge masses on rocks or in sand, and they reproduce by splitting themselves in half to create clones. So they can occur in large swaths. And it's also difficult to question, if one's mid-split, is that one or is that two? And so it's just a lot easier to count them as percent cover. And you can identify them with their green body and their pink and white tentacles, and they'll often be found on rocks or in sand. Next up, we have sponges. They're an encrusting species that you'll find on rocks, typically in the more sheltered sides of them. And you can kind of identify them by their open mouth-like structures called oscula. And they tend to be pretty variable in color, but easy to spot if you kind of know to look for their pores, which their phylum name is Porifera. So one fun reminder of how to identify them. Next up are bryozoans, and these are similar to sponges in how they encrust on surfaces like rocks, but they come in different textures and colors and forms. And you'll kind of see them ranging from a gelatinous to crusty texture. But one thing that sets them apart from the sponges is that they don't have the big prominent pores like a sponge. And they'll typically have more of this pattern to them, as you can see in some of the photos where it's kind of radiating. And again, similarly, we'll record for colonial ascidians, and these will be clustered, and they're made up of a lot of different individuals. You can kind of see it in the photos where the little colored cells, those are the individuals, and they're embedded in like a gelatinous matrix. So they're really squishy and kind of slimy when you touch them, and they vary a lot in color. And some other weird things, you might see egg masses or just things that you don't know how to categorize. And the best thing that you can do when you run into something that you don't know is, again, to reach out for help, review your ID guides, talk to a lead naturalist or staff person will help figure out where to categorize that thing that you found. And then it's also helpful to include things in the notes, which is located on the bottom of the second page, the backside of the data sheet. Yeah, so just continuing the rule of thumb of if you have questions, ask for help. And so I'll just briefly run through this. Erica went through some of the sediment categorization, but we also, in addition to doing it for our elevation profiles, we also record substrate for each quadrat. And so we'll do clay silt sand, which is just anything super fine grain, like sand or kind of a sticky mud, which we run into at Fidalgo Bay quite a bit. And then we also run into gravel and cobble, gravel being from about pea size to egg, cobble going from egg to basketball. And then we'll run into boulders, which typically you won't have a boulder in your quadrat, but sometimes it does happen if the profile line and the transect lines occur just at the right spots. And then we also have shell debris, which again is just if there's broken shells in there. And so here we'll go over a few strategies for recording percent cover, and then we'll run through some exciting practice quadrats, which were developed by the Port Townsend Marine Science Center AmeriCorps from last year. So big thank you to Megan for putting that together for us. All right, so this is an example where it's really easy to identify something based on just a quick visual. So for example, sometimes you'll have something really easy where you can just say, oh, about half of this quadrat is covered by ulva. So we'll put down it as 50%. And so this works really well for large sections, but that's not always necessarily what you'll see in the field. And so one tool that we have is called a chi-square, which represents 1% of the quadrat. And also in our quadrats, we have different cross-hatching lines that can be broken down into 1% squares within them. And so even though you might not have a chi-square with you, you can kind of visually break down and say, okay, part of this quadrat section is 1%, and visually just go from there. But typically we will try to have chi-squares available on site for the day. But this can be really helpful for when you're starting out and just trying to get an idea for how to visualize what 1% is, and when there's like smaller sections. So for example, you might not have a big 50% section, you might have like a 15 or 20. And so having a chi-square can help really with dealing with these smaller sections. And so here is an example of what we can possibly see. And I'll be throwing up a poll that you can use to test your percent cover skills. And so for this one, we'll be looking at what percent of the quadrat is taken up by green. This doesn't necessarily reference any particular quadrats that we see in the field, but this is just a fun example for testing our knowledge. And so I'll put yes, I can access the poll. So this is our first practice question. How much of the quadrat is covered by green? And the options are 14%, 24%, 20%, or 9%. And so I'll leave a little bit of time for folks to look things over. And also for reference, we have a 1% chi-square in the lower right-hand corner. And that's designed to be to scale for the image.
If you launch the poll, I'm not seeing it, but I just remembered I'm the host, so that might be why. Sorry.
Yeah, I'm seeing some people put in responses. We have 5 out of the 12 people sending things in. So I'll leave just a couple more seconds. Last call, 3, 2, 1, and I'm gonna launch. All right, so the answer for this quadrat was 14%, correct answer A. And some ways that you could maybe break this down, just to kind of provide a reference for folks, is so the 1% chi-square was really helpful. And you could maybe kind of estimate this large sort of diagonal section being roughly 10%. And then the little sections on the right being about 1%. And then the kind of longer skinny section on the left being maybe about 2%, which adds up to roughly 14. I think doing some of the percent cover can be really difficult, but it gets easier and easier with practice, and we always have people to help if you have questions. So that's our first practice. And then this one definitely is harder. So we'll need a little bit more time to go over it, but this is just an overall categorization of the different colors present. So I'll launch this poll real quick. See. Here we go. Is everybody seeing the poll number 2? Just a thumbs up is good enough. Awesome, thanks. Yeah, and so for this one, again, just a reminder for the 1% chi-square is a tool you can use, but review this slide for how you would categorize all of the colors, with answer A being 2% orange, 44% blue, 14% purple, 40% green. Answer B being 9% orange, 35% blue, 6% purple, 50% green. C being 8% orange, 40% blue, 10% purple, 49% green. And answer D being 4% orange, 40% blue, 10% purple, and 46% green. I'll leave a couple more seconds for folks to review the slide. And all right, I'll end the poll. Share results. And so answer D was correct, adds up to 100% and accurately reviews the quadrat. So thank you to everyone who participated with these. And next we're going to move into a real quadrat example. So if folks would want to review what percent cover the quadrat is in rockweed. This is a bit difficult because this is not as easy and clear cut as a little drawing. It's a picture of what you'll see in the real beaches. It can be kind of difficult. So I'll launch this poll real quick. And so what percent cover is rockweed? And the answers are 28%, 40%, 21%, or 37%. All right, I'll end the poll in just 3 seconds. 3, 2, 1, end. And so question A, or answer A, 28% is correct. And again, this is kind of hard to do, looking at the real examples, but if you have any questions, there will be plenty of folks to help on the beach. And next up, we're going to do individual counts with Eleanor.
Great, thanks, Wendy. One thing that I want to make sure that we cover that I'm not sure has been quite said yet, but when we set out the quadrats, we're just looking at like the aerial view. So you shouldn't be looking underneath rocks and things like that. So looking at this picture here, you can clearly see the sea cucumber that Randy's pointing at with the arrow up there, the orange sea cucumber. So we can clearly see it from this view, but it's underneath the rock, and you may or may not be able to see that if you had the quadrat on top of that rock. And so just be aware for things like that. That's a common way that people get over 100% is they start looking underneath the rock and they start counting up more than 100% that way. So next slide, please, Randy. And just real quick on the reminder on the QA/QC form to make sure, so now we're going to go into the countable individual species, but on the front side of your sheet, so you're going to be entering the data onto the back side, and you can do the percent cover and the individual counts at the same time, but then make sure that in between removing your ulva or coming back the eelgrass that you're also getting these quality control checks or QCs to make sure that what you have is correct, and that then you can get the thumbs up to go ahead and either remove the ulva or come back the eelgrass before you move on to the next step, and then another QC photo will need to be taken so that we can, if we have questions later on, we can actually go back and look at the photos to make sure that the data that we're entering makes sense. Next photo, please, or next slide, I guess. And so here's the back of the sheet, and just a reminder, so we are grouping species, so you don't have to figure out, you know, which species of limpet you're looking at. All limpets are limpets because those little guys are so tiny and it can be really hard to see, which is also a reminder for those of you who wear reading glasses. Some folks find it really helpful to bring their reading glasses out because it can help them see a little bit better some of those tinier little critters that might be harder to spot. And again, when in doubt, always consult a lead naturalist, and we do have those photos as backup too. So next slide. So individual count identification, next slide. And in that last photo there, you could see that Kim here is holding a ID guide. We don't use those same exact ID guides anymore, but you guys will have these handy dandy ID guides that have photos of organisms that have been taken at local beaches, along with how we group them for these surveys to hopefully make it a little bit easier so you guys don't have to memorize all that we're going through now. You're definitely welcome to go back through the recording and look at that, but you will have these handy guides in hand to help orient you with identifying stuff. But hopefully a lot of these identifications are pretty easy. A lot of people already know what like barnacles and limpets are, for example. So next slide. This is meant mostly as a reminder. And so amphipods are the first of the countable species. They're usually pretty small. They are laterally compressed. So I think of amphipods as they start with an A. And so if you took that A and you squished in either side, that's how they're compressed versus the isopods, which I'll get to next. And so with the amphipods, a lot of people, like a great example are the beach hoppers or sand fleas, as some people call them. Those are the amphipods. Next slide. And they hop around too. So something to be aware of is that when you first get to your quadrat that you're doing is pay attention for anything crawling or hopping out of your quadrat before you get a chance to really sit down and settle in, because you do want to try to count those if you can. So isopods are maybe somewhat similar to amphipods, but these guys are dorsoventrally flattened. So I think of isopods, they start with an I, and then if you just squash down that I, that's sort of the direction that these guys are compressed. They do have segments and they do have 2 pairs of antennae. Next slide. So here's just a comparison side by side. Amphipods, they're laterally compressed or the A squished in, and then isopods are dorsoventrally compressed or the I squished down. Next slide. hermit crabs. They have 2 claws. They can be kind of cute when they look out of their shells, or the thing to take into account for here with these guys is that oftentimes they make a home in an empty snail shell. So make sure that you're not counting them as snails, or vice versa. Also make sure that somebody's actually home and it's not an empty shell. And a note that they do tend to move fast. And so sometimes you need to take account for them when you first get to your quadrat. And next slide. Crabs, they have exoskeletons, so be careful not to step on them. They can be a little bit fragile in that way, especially if they have just molted. They also have a pair of claws, and they come in all different shapes and sizes and colors. Next slide. So while we're out there looking, we are not likely to find European green crab, the invasive species, while we're out at any of our sites, but it is always a possibility. And so as some of you may know, like we have something called like the green shore crab, which is not always green. So similarly, the European green crab is not always green. Also not to be confused with our green shore crab or our purple shore crab, which can look somewhat similar because they're roughly similar in size to some degree, especially when European green crab are on the younger side, and they can have about the same color. But some of the distinctive features here is that they have oftentimes their teeth at the sort of from their eye stalks out can be a little bit pointier than especially our green or our shore crab, and they have 5 of those that stick out. So if you find one of those, you need to let one of us know. We are not supposed to like kill them or remove them from the beach, but we do want to document them. Part of the reason for that is that WDFW a long time ago was expecting green crabs to show up. And so they asked people to send them in, and then before they knew it, they had thousands and thousands and thousands of native green shore crab stuffed into their freezers from well-intentioned people thinking they found European green crab, but they turned out to be shore crab. And I see that someone thinks that they saw one the other day. So my suggestion is if you do find one, please take a photo and take something in the photo for scale so that we can get a better idea of what size it is. Feel free to send it to myself, but the Washington Sea Grant crab team is a good outlet, or WDFW's invasive species, aquatic invasive species program is also another good one. And I can try to post links for that later. Next slide. Shrimp is another one that we occasionally find out there. They usually have that sharp bend, especially like the broken back shrimp in the abdomen just behind the legs. Next slide. And they tend to be like the ones that we find are usually pretty small, and some of them can be kind of on the transparent side too. Sorry. So something else that we will commonly find out there are snails. And so snails, I think most people are familiar with what snail shells typically look like. And like I mentioned with the hermit crabs, make sure that somebody is home, make sure that it is a snail and not a hermit crab. Also, I guess at some of our sites, especially Fidalgo Bay, we do come across occasionally bubble snails, which are really, I think, cool snails because they look like slugs, but if you touch them, you realize they have a hard shell underneath the on their backs, kind of underneath their skin. So they look like they just have this little bubble under their skin of a shell. So they're pretty neat. And you can see in the photo on the right-hand side, there's a bunch of eggs behind this snail. And so that's something that we put in the notes, but we don't necessarily count, but you can always ask if you're unsure. Next slide. Limpets are another one. These typically cling to rocks. They have these shield-like shells, and they're they can be difficult to spot because in a lot of, at least our surveys, they can be pretty tiny and they blend in really well with the rocks. So keep an eye out for these. Next slide. Sea slugs are really fun to find. They don't have a shell. They often have these feathery-like features on them, but we don't necessarily see them when they're out of the water. But they can be really fun when you do get to see them in the water. And oftentimes we do see their egg masses out there too, which is something else that we note, but we don't necessarily count. So again, when in doubt, ask one of your lead naturalists. Next slide. Bivalves, which include clams and oysters. And so these have 2 shells. Just a quick reminder that mussels are counted in the percent cover. These are, which also have 2 shells, but these are oftentimes buried, so it's not super common that we actually get to count them, but on occasion we do find them. One thing to definitely be aware of is that sometimes clams or oysters will die, and then they'll sort of fill up and there'll be a suction that forms to keep their shells closed. And so before counting it as a live critter in your quadrat, maybe try to pry it apart and see if it comes apart, whether it's live or dead. Don't try too hard because we don't want to kill them, but oftentimes if they're alive, they're pretty strong and it's really hard to just pry them apart with your hands. And yeah, next slide. Oh, sorry, go back. I remembered what I wanted to say, siphons. So sometimes you can actually see clam siphons sticking out. It's really difficult to identify these. There's a number of things that can look very similar. And so if we can confidently ID them, sometimes we will, and then sometimes we'll just make a note of it, depending. So ask if you think you see some clam siphons, whether or not you should count them. Next slide. Chitons, these guys are a lot of fun. They have these 8 plates. They also cling tight to rocks, similar to limpets, and they often camouflage really well. And so keep an eye out for them. Sometimes they'll kind of be covered with some algae and or mud or something on their backs. And so they can be a little bit tricky to see, but they're tons of fun to see when you do see them. Next slide. Ribbon worms, these guys are fun. They're round and have really soft bodies. So if you, I don't recommend that you pick them up because they are pretty fragile. They can start to split apart if you pick them up and you don't support them well enough. But they are usually pretty long and stringy and come in all kinds of fun colors, like usually purple and kind of bright red or orange. And they don't have any feet or segments either. Next slide. The flatworms usually are found on the underside of rocks. And so in our surveys, especially in the quadrats, we are not turning over rocks. So you're not likely to see these guys, but it is potentially possible. So they don't have segments. They're really flat too. Next slide. Then there's the polychaete segmented worms. So these worms have lots of segments and feet and usually kind of squirm around a bit depending on what kind they are. So there's like the pileworm and nerdaes that you might see out there. And then on the right hand side, there's some of the tube worms over there. And I see that there's a question about, do dead barnacles count as shell debris? I would say yes, generally they would count as shell debris because they can be substrate or they likely will turn into substrate sooner or later as they break off of the rock. Great question. Next slide. So there's the aggregating anemones in the percent cover that you might remember. Any other kind of anemone, however, we that live solitary lives, even though they might still kind of be found in clumps. So beware, just because you see a group of anemones together, that doesn't necessarily mean that they're aggregating anemones. But if they are non-aggregating anemones, we count them as individual counts. Next slide. Sea stars typically have 5 rays, but there are also like sunflower stars out there too. And so we count those. Again, sometimes they are found on top of rocks in other areas, but oftentimes they're kind of wedged and hidden. And so they don't tend to show up as much in our surveys. They'll be around our survey sites, and oftentimes they'll get noted in our species swaths, but you might not find them in the quadrats, but do keep an eye out for them. Next slide. There's also the brittle stars. So these have sort of more of a almost like a disc of a body with 5 points on it. And then they have these 5 long slender arms. We rarely find those, but we have a couple of times. So do keep your eyes out for them. We found a really tiny one out at Fidalgo Bay. I remember one year that was really cute. Next slide. Sea urchins are kind of these tiny little pincushions. Oftentimes we'll find them kind of like a light greenish color, kind of like the one in the photo here, but there are other varieties out there. Next slide. Sea cucumbers, again, usually these hide under rocks, but occasionally we do find them out. And so they're these kind of orange and red blobby things that, so you can see this one here is underwater, and so it has its gills out, so it's feeding and breathing while it's underwater. But oftentimes if you see it in your quadrat, it will likely have that all retracted in, but keep an eye out for those. Next slide. Fish, again, rarely show up, but occasionally they can. So that is something that we can mark on the data sheet. And I think we're almost through. Next slide. All right, so just a few quick reminders: go at your own pace, make sure that you get quality assurance and quality control checked, ask for help whenever you need it, and make sure that you're having fun out there. So with that, I think we'll open it up to any other questions. And I need a cough, excuse me. Allergies. I've been out in the field a lot. So does anybody have any questions? Otherwise, we'll stop the recording and we'll open up breakout rooms where folks from Skagit and Whatcom can maybe stay in the main room, and then if you're from Port Townsend, you can go into the breakout room, and that way you can have a little bit more of a targeted discussion for your specific area. But are there any more general questions in the meantime? Going once, going twice. All right, I'm going to stop recording.